All right, part 4 of unit or chapter 9, cellular respiration. We're going to be looking at fermentation, but before we get to fermentation, I did want to recap oxidative phosphorylation, which includes both the electron transport chain and chemiosmosis. Recall that in the electron transport chain, electron carriers, NAD and FAD, they drop off their electrons, NAD, one protein, sooner than FAD. NAD will pass through each of these proteins by transferring from one cytochrome to the next down the line. It will eventually link up with water or oxygen to form water. But as it passes through these proteins, it pumps hydrogen ions into the intramembrane space. This creates a hydrogen ion concentration gradient, which is then used to drive the production of ATP that we see going down on our screen right here. So it kind of passes through ATP synthase, and it forces or provides the energy needed for phosphates to tack onto ADP, recreating ATP. And the process continues. So to put some more numbers to the whole process now, starting with glycolysis and tying it to the citric acid cycle and the phosphorylation, we can see that glycolysis produces 2 pyruvates, which is then converted into acetyl-CoA. We have some electron carriers being formed as we go through these processes, ultimately being delivered to oxidative phosphorylation. The citric acid cycle takes the acetyl-CoA, creates a few more electron carriers ready to deliver electrons, as well as a little bit of ATP. So glycolysis produces 2 ATP, citric acid cycle produces 2 ATP, and then oxidative phosphorylation really cranks out the bulk of our ATP, which ranges between 26 and 28 ATP. So in total, all of this combined for cellular respiration creates 30 to 32 ATP per glucose molecule. So for each glucose molecule we start with here, we'll get about that many units of energy. This is a little bit different for fermentation, which we're going to start talking about here in a second. So fermentation can come in one of 2 types. There's alcohol fermentation and lactic acid fermentation. This process will result in the absence of oxygen, so this is an anaerobic reaction. It's similar to cellular respiration that it consists of glycolysis and also interactions between NAD becoming an electron acceptor and an electron donor. But NAD will be reused by glycolysis in that cycle that we're going to be looking at here. So the top part of our screen shows us alcoholic fermentation, and the bottom shows us lactic acid fermentation. Both of these are ways to produce ATP anaerobically or without oxygen. Glycolysis can occur itself with or without oxygen, but depending on if there is oxygen present or not, that will dictate if we move forward into anaerobic respiration or if we actually move into cellular respiration within the mitochondria. Remember that oxygen is highly electronegative, and that was used in cellular respiration as a strong pull on electrons. Well, in the case of fermentation, there isn't oxygen. So we would need something else with some electronegativity to pull on those electrons through the cycle to generate the ATP. This is commonly sulfate that acts as the electronegative electron puller. Fermentation does not use the electron transport chain. There's no oxygen. So instead it uses ATP substrate level phosphorylation, which we spoke about way at the beginning of this chapter. Substrate level phosphorylation is when ATP is made when an enzyme transfers a phosphate group from a substrate molecule to ATP. We see this occurring in the images above. The recall that glucose picks up phosphates during glycolysis, which is going on right here, which makes the phosphorylation of ADP, or addition of a phosphate, possible in both alcohol fermentation as well as lactic acid fermentation. So we see a phosphate phosphorylating ADP, so we get some ATP. We see phosphate phosphorylating ADP during glycolysis to create ATP. And once again, in both examples here, we start with glucose, they go through glycolysis, and they form two pyruvates. So taking a closer look at alcohol fermentation, pyruvate is produced from glycolysis, and it can be converted to ethanol in two steps, with the first releasing carbon dioxide. Alcohol fermentation by yeast is used in brewing, winemaking, and baking, and we can see in this image how ethanol would build up, and how the degeneration of the pyruvates would accept these electrons, allowing the NAD electron carrier to kind of go in a cyclical motion here between glycolysis and the creation of ethanol. What should be noted here is that for 1 molecule of glucose in fermentation, we're only getting 2 ATP. So 1 molecule of glucose goes in, and we get some ethanol, but only 2 ATP are produced. So that's a very, very small quantity of energy relative to cellular respiration, producing 30 to 32 molecules of ATP for each glucose. And in similar fashion, lactic acid fermentation, we take glucose, put into glycolysis, it picks up 2 ATP, forming it, and forms 2 pyruvates as well. The pyruvates then go back this way. We accept some of the electrons from it. We get NAD forming again, ready to go get more, and we have lactic acid building up inside of muscle cells. Notice that in this one, there is no release of carbon dioxide. Lactic acid fermentation by some fungi and bacteria can be used to make cheese and yogurt, as well as create some very painful muscle cells in our bodies when lactic acid fermentation is used to generate ATP when oxygen is scarce.